Hello, and welcome back to the Iceberg, where we delve a bit deeper into the wonders of neuroscience and the world of neurobiology. I'm Shivam Kill, your host, and today we're going to talk about something I find to be quite fascinating: optogenetics. Optogenetics is a technique that, unsurprisingly, combines both optics and genetics in a manner that I find to be quite interesting. And neurobiology finds it quite interesting, especially since how many different applications have been made using optogenetics. The main technique optogenetics employs in neurobiology is the use of a type of virus called adenoviruses to encode photosensitive proteins. Basically, for a quick speed breakdown, viruses are capable of making proteins using their RNA, which is basically like a different type of DNA. Maybe not a different type, it's ribose nucleic acid, but whatever. The process of doing so is complicated, and that itself would be saved for another day. But what you need to know is that these viruses can create proteins, which are basically capable of 90% of all functions in the cell. Now, these adenoviruses make photosensitive proteins, which, as it sounds, are proteins that react based on the presence of light, which means that light can be used to control the activation and deactivation of these proteins, which can in turn inhibit or activate other parts of the brain as well. Because this method is almost completely non-pervasive, since the only thing actually pervading or invading the brain is harmless viruses, and the fact that this is a super precise method, optogenetics has gained immense popularity overall in the scientific and neurobiology community. So how does it actually work? Well, we have to consider what we know about neurons. Neurons are a type of cell in the brain that communicate via electrical and chemical signals. They use ion channels to ionize their bodies, also called axons, and shoot an electrical pulse that cascades throughout their axon and activates other chemical messengers at the end of the neuron, the axon terminal, which will trigger another electrical pulse in the dendrites, basically the receivers of another neuron, and that electrical pulse will travel down its axon into its axon terminal and then continue with a chemical signal, which will so on and so forth extend forwards, creating basically a massive neural network that uses electrical signals to do desired tasks, whether it be moving a muscle or thinking about food or helping you fall asleep. So what these photosensitive proteins are basically capable of is controlling the specific neuronal signals within these massive neural networks, either shutting down the entire pulse or empowering it as a whole. And this ability of these photosensitive proteins is what scientists absolutely adore about optogenetics. Let's assume, hypothetically, that some neurons are contributing to a disease. Let's say, in this example, that there are some neurons in the motor cortex, for example, that are causing your arms to constantly windmill without stopping. What optogenetics can do is that scientists can embed these photosensitive proteins within specific neurons, in this case, the neurons that are causing these windmill patterns. And with the mere shine of light or triggering of these photosensitive proteins, the signal gets suddenly interrupted, and out of nowhere, the arms return back to normal. Now, this is a very, very oversimplified analogy for how this works, but it's the basic methods of how optogenetics can contribute to the field of neurobiology and the potential that comes with the ability to control the signaling behavior of certain neurons in a vast neural network. Speaking of that, we should also note that the specificity of these proteins is what makes this method so powerful. If scientists are able to control specific neurons in the brain, rather than have to worry about the entire neural network as a whole, they're able to effectively and precisely deal with the cause of certain diseases by targeting the malfunctioning neurons. Speaking of this type of research, most optogenetic research has actually entered into the clinical trial stages which means they are now testing it on actual patients with neurological disorders, which by itself is a huge step for the presence of optogenetics in the medical field as a whole. Now, some interesting results in the preclinical trials, which are basically the trials before we get into the human trials. Bless me. Sorry about that. And that should also be discussed because some of them are actually quite fascinating to look at. For example, in a research on optogenetics in mice, there are many, by the way, that are coming up. A group of researchers implanted light-sensitive proteins within the hippocampus of these mice. Basically, the hippocampus is the main producer of memories in the brain. So it's kind of important to how you're able to remember things, anything. So, you might want to deal with that if you're having memory issues. And what they found was that when exposed to light, the mice were actually able to activate memories of themselves. And without the light, the next day the mice lost these memories. Basically, the connection between memories and processing that most Alzheimer's patients don't have are effectively connected with the use of optogenetics and photosensitive treatments. Further developments into optogenetics may yield some fascinating results pertaining to potential Alzheimer cures, though we should probably recognize that most of these results are still only applicable to mice and still need to be tested in an actual clinical setting with real human patients. There's actually another study, if I remember correctly, where optogenetics was used to trigger certain neurons in the brain that assisted in Alzheimer's alleviation, though the specifics of that study I fail to recall. Moving on, in Parkinson's disease, another neurodegenerative disorder, apologies, it's quite late where I am. We see a potential treatment with the usage of optogenetics, something that many researchers in Parkinson's are very focused on at the moment. Our research dedicated to deep brain stimulation, which is actually something we've talked about in a prior episode, with the usage of photogenic triggers, actually showed some very significant alleviations in the symptoms of Parkinson's in mice. In big words, they combined optogenetics with already successful deep brain stimulation methods to effectively find some ways to alleviate symptoms of Parkinson's, most of which are related to tremors or motor dysfunctions. And obviously, all are nice. We're not doing clinical trials as of yet because we're discussing preclinical, obviously, and also because post-clinical or like clinical trials are a bit more messier to handle because they're humans. Anyways, what was I? Oh, right. They're doing research with deep brain stimulation using optogenetics. This research, for example, was mostly focused in the axons of the subthalamic nucleus. Big words, I know. Axons, if you remember, are like the body of the neuron. It's what holds that electrical signal. The subthalamic nucleus is basically a hub for inhibitory movements, like the thing you want to have when you have uncontrollable tremors. Uh, Basically, inhibitory, again, opposite of excitatory, it will stop something from happening. So if you have too much movement because of these tremors or motor dysfunctions, inhibitory things would stop that from happening. And this sort of treatment with optogenetics was actually able to significantly treat Parkinson's disease, especially the many motor deficits caused by it. Lastly, even in the field of vision, funny pun, I know, optogenetics are showing some amazing results, especially pertaining to color blindness. Researchers basically sensitize the cell to light by encoding proteins that encode, again, light-activated channels and pumps. Not mechanically, they're basically just fancy words for light-activated ion channels or pumps, as in like you pump in ions. Most of biology relies on ion pumps or chemical gradients or whatnot. This basically means that by using the viruses, the ones that we mentioned in the beginning, scientists are actually able to make proteins in the eyes more sensitive to light. And with these photosensitive proteins are actually able to perform different functions as a result of light activation. which is how they're able to alleviate color blindness. The sensitive proteins are going to like trigger some sort of site that does that. I believe it's something to do with the no, yeah, the cones side of the eye, which are basically part of the eye, just rods and cones. But ignore that. I'm not going to go too much into specifics because this is just a general overview of optogenetics. There's some research going on because you're talking about color blindness. There's also research on treating blindness as a whole, but that's still very much in the ground stage because one of the biggest steps in that field so far has only managed to get pure blind mice to partially be able to see, which, by the way, is still like an immense step that is going from no sight to having sight, even if it's not the best. But it's not one that's big enough to apply to humans in trials or even in mice yet. But maybe with the further research in optogenetics, we'll see a change in that sort of experiment soon. Anyways, I hope you all learned about the importance in this field of optogenetics and how it can be used to define the future of medicine one day, which again is a bit of a misleading statement since optogenetics already is defining the future of medicine. So I guess I'm kind of off there. Yeah, so this is Jay Kell, signing off and keep diving. Peace out.